So this video in our course B01 in Data Management includes a discussion of spreadsheet data, which is a more fundamental than high-level discussion of how we manage our data. So as we've been discussing already, we want to keep things simple and systematic and easy to understand. So we have our columns are our variables or features, and our rows are our observations, typically our samples or whatever we're looking at. And your goal is to be simple and accurate and clear, not creative. You don't need to do things your own way, just because you're trying to show that there are other ways of doing things. So we should have one variable per column. If you need more columns, you can add them. That's not an issue. One variable per column, and the column should only contain one data type, so text or number or whatever. Another important point is that you don't do calculations outside your spreadsheet. You have your raw data separate from your process data. So it's trying to create multiple data sets where you separate your raw data from your process data or data you're trying to analyze. Ideally, you use code, but that may not always be possible. But think of this is that you don't want to pee in your own bath. So you don't perform your analysis in the same spreadsheet as where you have your raw data. So for example, you see examples here where you have different variables for different samples. You can see there's a binary data, continuous numerical data, labels here. And you see then we have issues that if we have, say, multiple variables inside the same cell, multiple times the information inside the same cell, we can actually separate them on. So we can separate here to have a replicate column as well as a variable 3 column determining if they're wild type or infected. And that can make the data more easy to understand. We can even remove the term replicate here from these ones and just keep that as numerical data itself. So there's lots of resources that I'm pointing to here that will help you achieve this. For example, wherever possible, store data in one sheet. Don't split it across multiple sheets. Include a data dictionary or a code book. If you're dealing with human data, de-identify it. Do not merge cells. That means it's not machine readable. If you have blank rows or columns, remove them. And don't do calculations inside your spreadsheet. Raw data or cleaned data should not contain calculations. So for example, here, how not to do it. You see the first column is empty. The first few rows here are empty. These are more empty rows. Software tools will not be able to process them. You've got 2 different tables in this data set when there should be 2 different spreadsheets. And you have the relevant metadata recorded inside the same spreadsheet, whereas that should be in a separate spreadsheet. So more things that missing values should be recorded as NA, not left blank. You should ensure that the formatting of headers is consistent. Don't include numbers and text in the same column. Use consistent names that are linked to a data dictionary. If you say, for example, you're making a measurement where you have 2 different values that are linked to each other, referring to a single variable like blood pressure, you can split these into 2 columns. And that means it's much more easy to manage. And finally, don't use hyphens at all in your data set ever, because Microsoft Excel will just mess those up. So fundamental things that I may have mentioned already, use numerical codes, not alphabetical codes. So 1, 2, 3 is better than ABC. If you have relevant metadata comments, put them in your data dictionary, not inside the spreadsheet itself. Use consistent formatting for dates, which we mentioned already in this course. Ensure that your conventions are the same within a column, so it's using the same units. Do not write the unit inside the cell, put it in the data dictionary. For dates, because of the way that spreadsheets function, it's best to use separate columns for a year, month, and a day. A further nuance to this, that if we move from dealing with a single spreadsheet to dealing with multiple spreadsheets, is that we might want to segment our data. So basically have it across multiple spreadsheets. So we may have different data types that are present. For example, we might have a list information on a set of farms, and each of the farms contain information on their plots, which are separate on the plants that are contained inside the plots. So we might have 3 levels of information. I might have identifiers that bring us from the farm to the plots, and then identifiers from the plots to the plants. For example, you might have mutation data as linked to mRNA data, and as identifiers linking the two to allow us to link the two together. For example, here we have transcriptomic data, genomic data, cytometry data, and proteomic data. I might have samples and identifiers that are linked from one data set to another so that we can link things across different components. So consistent structure is very, very useful. So dates recorded in this manner will be more easily understood if they're a consistent way. Columns headers should be short, clear, and descriptive columns with no symbols or spaces in alphabetical characters only. They should contain a single data type, such as the sample, location, date, species. You can see there's no spaces or anything like that, like this. Normalization. So use unique identifiers to link different data sets across multiple sheets rather than duplicating data. So if you find yourself looking at spreadsheets that are like the following page, you know that you're dealing with something that is really, really challenging to deal with. So for example, we might have a good name here where we see there's no spaces, there's just underscores inside there. And this is a bad example where you see the units are inside the name with strange symbols and spaces. You got a good name is one that's long and easily understood, where this is a bad name because it's just not clear what that refers to. Another example is where you have a clear name, which is separate from one that contains a peculiar symbol that might confuse people, or maybe a short name that's not clear, whereas a long name will be more easily interpretable. So how not to do it? So for example, you see spaces, symbols, units, and label meanings inside there. This is bad practice, and we just confuse people and mean that is not machine readable. So that's why we need to want to separate the data dictionaries and the metadata separate from the data itself. So cleaning is something we're going to talk about in detail. So you might have spreadsheets containing data like this, where you may have some missing values, or they're recorded as NA, so there's no information available. So for example, it may be relevant for us to remove duplicates to ensure the data is integrity, or at least we should check for that. Missing values are, unless you're using methods that specifically will infer missing values, you shouldn't be trying to guess them. It should be recorded as NA, not zeros, nor even the cell lengths or any other value. NA is the way of recording that. And then you should consider for your area what data validation rules do you have. And we'll do this with OR in the lab. And ways of doing that will be ensuring that your good data is positive, or the dates are valid, for example. Another key thing is think that raw data is the primary data that we deal with. Processed data is something that that's been processed somehow. So these are the raw files for microarray analysis, CEL files. They contain information like this. We might extract from that information, say, the fold changes across different genes or transcripts for comparison. Well, what's worth thinking about is that the processed data is not the same as the raw data. So the processed data is a bit like cooking, that this is irreversible, and that we want to share the raw data so people can repeat the same process of cooking. Or if we're only sharing the process data, it's not very useful for the community. So in the same way, when you're cleaning data, you should separate the raw data from the clean data. You may have multiple steps, and therefore you may have multiple stages there that we're going through multiple cleaning steps, and you have multiple data sets that bring you towards your cleaned data, and that's fine. That will allow you to validate your data more effectively than try to do it all at once. And you may have separate analysis, and therefore you may have separate data sets that are tightly controlled and consistent with each other, but are analyzed in a different way. And What will help you achieve that is if you've a consistent data dictionary, a way of naming things, create a data cleaning plan using the data dictionary. What issues could your data have? For example, there could be missing data. There could be data where you have extreme values that you shouldn't have for certain measures. You may want to remove data that's uninformative. And what we'll do in the lab part is import the data sets into R. So you'll bring your own data and import it into R and visualize things for it there and use their commands to check the integrity of your data set. And the advantage of this is that using code will mean that it's more easy to identify errors such as this that occur all the time, such as using uppercase and lowercase. You've got space there with sunny spells. All these things do occur. O's and zeros is another example, ones and L's. There are lots of ways in which data sets may be not as ideal as we think. We also want to make our data set to be machine readable wherever possible. And that's different from machine actionable and machine interpretable. So we're used to machine readable as being something like a structured data set that's also readable by humans. We may have machine actionable files. These may not be machine readable. So they may be semied or unstructured files that only a computer can interpret. So for example, image data or OS data often can be in that particular format. And what we want to avoid is data sets, recording our data sets or storing our data sets or even publishing our data sets in PDF format because that's human readable but not machine readable. And you see a lot of journals in particular publishing things as PDF, which is publishing data sets in PDF format, which means it's really challenging to extract that information. So machine readable data, we should try to avoid merging cells. Symbols, never use non-alphabetical symbols. That's not a data convention. Don't use colors at a column containing a classification and use consistent data types. So each column contains one type of information, one type of data, either numerical text or whatever, and use standard coding ways that are universally used. And think about this, that if you're using colors, then it's just easy to add on another column. So for example, if you look in different papers, you can see some examples for 86 pages of full changes inside a PDF. How on earth is that supposed to be useful for others? Or you might have image data or numerical data inside other PDFs or images. It's not very useful. In the same way, non-human readable meta-analysis may look like this, where we have this kind of spreadsheet where you have time here with space and symbols, and then we have numerical data, you've got highlighted cells, and this is the type of thing where it contains calculations. This is the type of thing we want to avoid. So what we want to do is have one column is one variable, one row is one sample, one cell contains one piece of information. The first row is a header. In the same way, the first column contains information about the samples, and we have no special characters. So this means we can create tables that are tidied like this, where you have maybe the strain, then the genotype, normal and mutant, the minimum value you've recorded, and the replicate that is present, and the response. So that's a tidy data format that's simple and easy to understand. Whereas if we've got spreadsheets like this, where we have this is a bit of a mess, it might be interpretable for the person who is doing this, but this is not very useful and doesn't use a systematic way of doing it. If we have complex projects, we should use version control, and this can track changes to data sets over time, which can be good if we're dealing with a data sets that's spanning several years. So we mentioned here about different types of data sets. We're used to structured data being wide data, where we have rows or observations or samples and columns of the variables. So we might have blood and muscle readings before and after for a particular person. You can see that this is not formatted in the way that we want. And this is for 2 people. So what I'm going to do using R is convert this into long data. So each row has a single numerical value associated with that, and it contains multiple columns with the metadata. So for example, you might instead have the sample, the time, type, and then the measurement associated with that. So that's long data. So you can see one value per row. And therefore, if we transform Y data into this format, you'll have a very large data set, a very long data set. There's another example where you might have the group and the sex and the sample and one measurement associated with that. So that concludes our part on spreadsheet data.